Welcome to the Department of Electrical and Computer Engineering Distinguished Lecture Series. This lecture is also co-sponsored by the University Committee on Lectures. We are very privileged and honored to have Dr. Leroy Hood with us today. I guess the best introduction that I can give to him is that we had to delay the presentation for 10 minutes to put more chairs to accommodate the people who are flowing in. And I just watched the number of chairs in the room double over the last 10 minutes. Dr. Leroy Hood is the president and co-founder of the Institute for Systems Biology. In the past, he served as the William Gates Chair of the Department of Molecular Biotechnology at the University of Washington. He made many outstanding contributions throughout his career in biotechnology, systems biology, medicine, and bringing engineering to medicine. He led the biotechnology revolution from the front by inventing 5 pivotal instruments related to DNA, RNA, and protein sequencing, including the DNA sequencer, which enabled the Human Genome Project. He cured the first neurological disease by gene transfer in mice, and he played a role in founding more than 14 biotechnology companies, including such highly successful and large companies as Amgen and Applied Biosystems. Dr. Hood has published over 680 papers, received 26 patents, and 17 honorary degrees. He co-authored several textbooks in biochemistry, immunology, molecular biology, and genetics, and he is currently finishing up a book on systems biology. He co-authored a popular book titled The Code of Codes on the Human Genome Project. It's on sale in that corner. Although I haven't asked Dr. Hood for permission, I'm sure we can trick him into autographing books if you like. He's one of only 7 scientists in the United States to be simultaneously elected to 3 national academies: the National Academy of Science, the National Academy of Engineering, and the Institute of Medicine. Dr. Hood is currently focusing his energies on creating a paradigm shift in medicine to what he calls P4 medicine: predictive, preventive, personalized, and participatory. He recently co-founded a company called Integrated Diagnostics to create a platform company for this paradigm shift. And he will be talking to us about it today. And before I forget, this talk will be followed by a reception. So please stay back, ask questions, and you'll have an opportunity to interact with Dr. Hood. Please join me in welcoming Dr. Hood to Iowa State.
Thank you very much. Well, thank you for that gracious introduction. I'd like to take you on a whirlwind tour of where I think medicine is going to be going in the future. And I'll talk about the origins of the change, the technologies of the change, the role systems biology has played in the change. But most especially, I want to get across the idea that this new medicine is really predicated on the idea that medicine is an informational science. And to give you an example, my prediction is in 10 years or so, each individual patient will be surrounded by a virtual data cloud of billions of data points, and we'll have the IT for healthcare to reduce that enormous data dimensionality to simple hypotheses about health and disease. So what I'd like to do then is talk a little bit about the framework for this revolution, the idea of complexity and the paradigm changes that led to it. I'd like to talk about the fundamental pillars of P4 medicine, and then really what P4 medicine is, its societal implications, and then how are we actually going to bring P4 medicine to patients? What are going to be the major challenges? When I was thinking about the 21st century, 10 years ago, 15 years ago, it was clear to me at that time that the fundamental challenge for all of the scientific and engineering disciplines was the challenge of complexity. And what I became convinced of is that biology is uniquely positioned to deal with its complexity because of this idea of biology and medicine being informational in nature, systems approaches, emerging technologies, and new analytic tools. What's important about this supposition is this gives biology the wherewithal to attack some of the most fundamental problems in society, not just healthcare, which I'm going to talk about, but global health, the environment, energy, nutrition, agriculture, and the like. So I'll argue that the paradigm changes and approaches I've laid out here could be applied equally well to any of those other areas as we move ahead. When I was a young assistant professor at Caltech back in 1970 or so, I read this book by Thomas Kuhn on the structure of scientific revolutions, and it's really about paradigm changes, and it's about arguing. He discussed what paradigm changes were and how they were brought about. And the reason I became so fascinated is it was clear to me, and it turned out to be true, that I wanted to participate in a number of different paradigm changes. And I'll tell you about 4 paradigm changes I've participated in that really have fundamentally laid the framework for the 5th paradigm change, which we'll talk about today, which is P4 medicine. So the first paradigm change had to do with bringing engineering to biology, and that was the development of lots of technologies that you've already heard about. The second came about as a consequence of developing the automated DNA sequencer. Namely, in the spring of 1985, I was invited to the first meeting that was ever held on the Human Genome Project, and it became obvious to me at that time that this was a transformational and revolutionary opportunity for biology. What came from the Santa Cruz meeting, where 12 relevant scientists were asked to come and talk about the Genome Project, were 2 interesting facts. One, that it was technically possible, although at that time certainly difficult. And 2, there was an enormous split, 50 to 50, and the 50% that were against the Genome Project were really vehemently against it. And in fact, when you went out in the community at that time, the split was probably 90 to 10, at least initially. And one of the most dominant organizations that was against the Genome Project until the very end was the National Institutes of Health, something that people don't remember. A National Academy Committee I was a member of that had half opponents and half proponents actually gave a unanimous endorsement in 1988, and that really turned the tide. And NIH quickly saw that this was an opportunity rather than a challenge. What came from developing the automated sequencer was the realization that we had to bring into biology all sorts of different kinds of scientists. And I went to the chair of the president of Caltech in about 1988, 87, and I proposed I create a cross-disciplinary biology department there, and the biologists vehemently opposed it. And in 92, it was possible, with Bill Gates' help, to actually move to the University of Washington and set up the first cross-disciplinary department. And this department was incredibly successful. It invented the first fundamental technologies of proteomics. It pioneered the software that fueled the Genome Project with regard to assembly and quality verification. We there invented the inkjet printer that is what Agilent uses today to make DNA arrays and synthesize long DNA strands and so forth. We had a whole series of different things. But it was obvious even at that time that what I really wanted to do was, in fact, systems biology. And what I became convinced of is the bureaucratic constraints of a state university made it impossible to do many of the things that were required to really realize systems biology. So I resigned from the university in 2000 and started this Institute for Systems Biology, and we'll talk a little bit more about that in just a few moments. So it was the integration and synthesis of these paradigm changes that led to the final paradigm change that is the subject of this lecture. Now, I would make a number of observations about these paradigm changes. So observation number one is that they really fundamentally changed how either we thought about biology or certainly how we did biology. Observation number two was it's unbelievable how conservative most scientists are and how much difficulty they have in thinking outside the box. And this is a topic I'm going to return to at the end of my lecture. And number three was the really interesting idea that for every one of these ideas, we had to create new organizations in order to realize them. And again, we'll talk more about that at the end of the lecture. So the four fundamental pillars of systems biology, P4 medicine, are exactly what we enumerated earlier. The idea that biology is an informational science, that these systems approaches mandate that we approach the complexity of biology in a holistic manner, that we have to invent new technologies that will let us explore new dimensions of patient data space. And finally, we need the analytic tools to be able to handle all of this information. So what I'd like to do is talk about each of the four of these and give you an idea of how we've moved forward either in thinking about these and or in developing new approaches and or technologies to mediate them. So let's begin by talking about biology as an informational science. And I would argue there are really 5 fundamental ideas that you have to understand in this context if you're really to be able to do systems biology. So idea number one is the idea that evolution is not directed, rather it builds on random mutations and environmental conditions to shape unique solutions to various types of problems. And the really key point is it doesn't use the law of Occam's razor. It's much more evolution enacts in a Rube Goldberg apparatus, because evolution builds upon what went before. And in fact, the well-known fact, there are a series of insects called the stick insects that evolved and de-evolved wings 3 or 4 different times. And with each of those events, there was recorded in the genome information, and it led to a Rube Goldberg-like kind of machine. So for those of you who are engineers and computer scientists and theoretical types, the only way to deconvolute the complexity of biology is to start with the data itself. That is the only way we're going to gain deep insights into the data. Second fundamental point is modularity is really the key to being able to understand complexity and to scale the different levels of biological complexity. So all of this is obviously made by Legos, and you can see different entities here that have very different functions. And once you understand the basic Legos, you can then use them to build structures. Once you understand the structures, then you know what all the related structures might be doing, and so it goes on with biology. There are fundamental modules that can be used in many, many different ways that solve fundamental information processing problems. And by coming to understand these fundamental modules, again, it's a real key to deconvoluting complexity. There are fundamentally 2 types of biological information. There's the digital information of the genome. There are the environmental signals that come from outside the genome. And it's the collision of these 2 types of information that create the 3 processes of life, or 4. Evolution, development, physiologic response, and disease. And we have to understand these processes then in the context of what are the relative contributions of these 2 fundamental types of information. A second point that is really important is how do you connect those 2 types of information with the phenotype, the appearance of the organism? And we do that by 2 types of informational structures. One are biological networks, which capture and transmit and integrate and mine and ultimately pass off information to the second type of structure, complex and simple molecular machines. And the interface between these two is not always obvious, but these lie at the heart of what systems biology is. Understanding their dynamics, both from a temporal point of view and from a spatial point of view, is absolutely critical. Now, to give you an idea about relative contributions to phenotype of environment and genome, these are the left index fingerprints of two identical twins, and you can see they're absolutely different from one another. So this means, if they have the same genes, and they do more or less, that the differences are environmental. And the question that is absolutely fascinating is, one, do the differences rise as a consequence of stochastic events that occur during the differentiation process of the fingerprints, or do they occur as a result of stochastic events that come in from the environment and modify the output of the fingerprint program and everything? We don't really know the answer to that. And the final point I would make about information is that it is multi-scale, it is hierarchical, and it is absolutely critical if you're to understand a particular system at one level, say the single cell and how it divides. You have to capture all of the antecedent levels of information right back to the genome, because the environment impinges upon and modifies every single one of these levels of information. So we have to be able to capture the information at levels and carry out an integration process which operationally explicates the environmental contributions at each level to the digital signal. And we don't know how to do this very well at all. We're still in the process of learning. It's a terrific problem to think about for students and so forth. So these are these 5 fundamental principles, and I'd argue they are absolutely essential to really thinking in a deep way about systems biology. Okay, let's talk about systems approaches to biology and what that actually means. So my view of systems biology, and I'm going to give you a simple analogy because I really got invited by an engineering department. So my question is, suppose you were an engineer and you wanted to figure out how a radio converted radio waves into sound waves. The first thing you do, obviously, is you'd take it apart and you'd look at its individual components and maybe come to understand what those individual components could do. And this, of course, is what biology has done primarily for the past 40 years or so, looking at individual genes and individual proteins. What the Genome Project brought in the way of a revolution was a complete parts list, and for the first time we could do coherent systems approaches to the understanding of biological complexity. The second thing the engineer would do, obviously, is assemble those parts back into their circuits and then come to understand individually and collectively how these circuits converted electromagnetic waves to sound waves. And that's exactly what we do in biology. The environment and the digital genome impinge on the human and biological circuitry, gives a phenotypic response, normal and or diseased. And if you think about systems biology, I would say there are really fundamentally several really important components. Number one is the idea that you take all of the information about a complex system you have and you formulate a model which can be descriptive, graphical, and or mathematical if you have enough information. And then you formulate hypotheses to test that model, and you explicate those hypotheses by perturbations of the system. You integrate the data from the system's perturbation back into the model, changing the model in accordance with the reality that's been revealed by those data. And you do this in an iterative fashion until you bring into proximity experimental data with the model itself. And the ultimate idea of what we want to be able to do is to generate models that are predictive and actionable. So actionable means you can actually tell a patient something that will be useful for them to do in improving their health. And we'll talk a lot about that a little bit later on. Systems biology has a very rigid set of views with regard to data. Number one, it argues wherever possible you should do global data acquisition because you never know which data is really important. We can do that in genomics. We can't do it in proteomics and metabolomics yet, although in proteomics we're beginning to get close. You have to be able to integrate the different data types. I've already talked about that. You have to be able to delineate the network dynamics, molecular machine dynamics, and again do it in both temporal and spatial sense. And finally, and this is really key with large data sets, most of what you look that varies normal to experimental is noise. How do you deal with the enormous noise in large data sets? One of the biggest fundamental problems in biology. And I'll tell you there are two types of noise. One type is technical and the other is biological. Because if you measure any aspect of phenotype, often it is the sum of several different biological phenomena. And if you're only interested in one of them, you have to subtract out the others. So how do we do that? And I'll tell you one way we can go about doing that later on. And the final point, of course, is discovery science, that is being able to enumerate all the objects in a biological entity so you have a complete parts list is absolutely key to systems biology. We're struggling to do that now with proteomics and metabolomics and so forth. What we've really predicated our approach to systems biology on is the idea that biology is the driver of technology, and that in turn is the driver of computational and mathematical tools. And the pace has to be set by the biology. And we've created a real cross-disciplinary culture. And there are two things you have to be able to do with cross-disciplinary cultures. One, you've got to teach people to learn to speak the languages of the other disciplines. And two, you have to learn to work together effectively in teams to take on big problems. And actually, three, you have to learn how to educate new students to be cross-disciplinary. And that's going to be really important in the future. And if you can do that with that philosophy, you can generate then information that's useful in the context of the cross-disciplinary environment you see there on the lower right-hand corner of this slide. What we've done at ISB as well is we've created a lot of high-throughput facilities that any scientist has access to, as well as the bioinformatics and computational resources to be able to analyze that information and then apply it to simple and complex model systems and ultimately to human organs. So ISB is now 10 years old. It has 11 faculty and about 300 staff. And a digression for just a moment, it's always interesting in 10th anniversaries to think about metrics of success. And we actually have two really interesting metrics of success. Let me say, when I started the Institute of Systems Biology some 10 years ago, there was enormous skepticism and the proclamation that systems biology was all real hype. And what has happened that's been very interesting is last year a report came out from the National Academy called The New Biology for the 21st Century, and it's a perfect description of the systems biology that I've told you about here. And in fact, it argues that this is going to be the future of biology and medicine. And what's interesting is two of my friends that were the biggest skeptics about systems biology wrote this report, were on the report. So people have come around to a different way of thinking. I asked one of them, I said, why do you call it the new biology, which is terribly vague? Why didn't you call it systems biology? And they said, we didn't want to give the Institute too much credit. So anyway. Now the second metric is a personal one, and for us it's been absolutely spectacular. And it arose as a consequence of a Spanish institute that determined a new way of assessing impact factors on individual papers and then assessing over a 5-year period all of the papers that a particular institute had created and then giving an average impact factor for that time. And they looked at 2,200 institutes, and these are in all fields of science. And by that measure, the institute came out first in the US and third in the world in terms of the impact of its papers. And one of the really important observations was that we had high impact papers in biology, in medicine, in computation, in mathematics, and in technology. So it belies the power, it underlines the power of these systems approaches. Now, a systems view of disease in this network-centric view of systems biology I've given you means that in the relevant diseased organ, one or more networks becomes disease-perturbed, either in terms of genetics or in terms of environment. That alters the structure of the network, that alters the envelope of information that it dynamically expresses. And the argument is that dynamically changed envelope of information explains the pathophysiology of the disease and gives us brand new insights into diagnosis, into therapy, and ultimately into prevention. And I'm going to show you with one example that all of this is true. And the example we're going to talk about is a neurodegenerative disease in mice, prion infection, that we've studied for the past 7 or 8 years together with George Carlson at the McLaughlin Institute. And the essence of this disease is absolutely fascinating. It arises from a protein, a normal protein, who can have its three-dimensional structure altered to become, quote, infectious. Once the infectious protein arises, it has two interesting properties. One, it can catalyze the conversion of the normal to the prion infectious form, so the disease is autocatalytic. And this is actually one of the most dangerous infectious diseases. It's the disease that no neurosurgeon ever wants to touch, and usually they throw the tools away. They don't even try and sterilize them. And the second thing is that infectious protein is utterly resistant to all known proteases, and almost certainly that's a part of the basis for the reason it aggregates in neuronal cells and kills them and leads to the neurodegenerative phenomena. But for us, the reason that it was a nice model system is if you infected normal animals with these infectious particles, you could time precisely the beginning of the disease process and follow it through to its termination. So you could do dynamical studies all the way across disease progression. And that's exactly what we did with several inbred strains of mice. The idea was to infect one strain and then to look at normal counterparts. We analyzed the brain transcriptomes using DNA array technology that let us visualize most of the genes. And the most important thing that we did was at each of the 10 time points or so, subtract the disease transcriptome from the normal transcriptome to get genes that differed. And of course, we were horrified to find that initially 7,400 genes differed. That's more than a quarter of the mouse genes differed one from another. So the idea was that there is enormous noise in those 7,400 genes, and the question is, how do you get rid of it? So what we did was to infect 10 different prion strain, inbred strain combinations that each exhibited different aspects of biology that we could subtract away from the fundamental biology we were interested in, namely the genes that encoded this neurodegenerative prion response. And to give you an example, one of the animals we looked at was a double knockout for the prion gene. It never got the disease when infected with infectious prions, so any changes in its brain transcriptome could be subtracted away. They were irrelevant to the disease process. And so we were able to, with those 8 subtractions, get down to about 300 differentially expressed genes. So that was more than a 20-fold increase in signal to noise. So the really interesting question is, what do we do with those 300 genes now? And the idea was to take the 4 most fundamental processes in in prion disease, and to look at the networks to the extent we knew them, and then to map onto each of those 4 networks those 333 genes that actually changed across the 10 time points in the progression of the disease. So we looked at these differentially expressed things for all 4 of these networks and made a series of really key observations. So the first observation that was fascinating is the 4 major networks were sequentially disease perturbed in an order that never changed. Prion accumulation first, and then glial activation, and then 2 different forms of neuronal degeneration. So it means there's a sequential process, and if you're interested in therapy, you want to focus it on using drugs that can make the initial network early on behave in a more normal fashion to abrogate the entire disease process. And this is the strategy that will revolutionize how drug targets are chosen, and we're beginning to test this in yeast as a model organism at this point in time. The second thing is, if you looked at the dynamics of how this is the prion accumulation network actually changed, you could come to a number of absolutely fascinating conclusions. And remember, we looked not only at this network, but at 3 other major networks, and as you'll see in just a moment, we looked at some other ones. So the major conclusions were, 1, we could say about 300 genes were involved in this process. Number 2, we could say about 2/3 of those genes mapped into the 4 major networks, but the other third mapped into 6 other networks that nobody had any idea were involved in this disease process. And with all of them, we could see how it made a lot of sense for them to be involved. Finally, as I've already mentioned, the networks were disease perturbed in a sequential fashion. But the really important point is, collectively, these network changes explain every aspect of the pathophysiology of this disease that we knew for the first time ever. It also opened up new ways of thinking about drug target discovery, as I already enumerated, and it got us thinking about a completely new approach to diagnostics, an approach where we will make blood a window into viewing health and disease for the individual. And the approach that we took to doing that was really twofold. One was to look at those networks and get key changed nodal points and ask, were they secreted into the blood? And if so, were their concentrations altered in accordance with their transcript concentrations? And with many that we looked at, the answer to that was yes. So that let us, for the first time, do beautiful pre-symptomatic diagnosis of a disease long before any clinical signs ever showed up in these animals. And I think the second approach we're taking is going to turn out to be maybe the most important, and that is we could use deep transcriptome analyses of 40 organs in humans and mice to identify transcripts that were uniquely or relatively uniquely expressed in each organ, and then ask, were those secreted into the blood? And of course, if they were secreted into the blood, then that meant that created a fingerprint of, say in the case of the blue, brain-specific proteins. And the idea is that those fingerprints in the blood then will record reflections from the disease process that occurs in the brain. And each of the proteins in the brain-specific fingerprint, and we've now identified for human and mice more than 100 such proteins, okay? Each of those proteins reflect the operation of a particular cognate network. So if the brain is normal, the 100 levels will be set at one thermostat stage. And if there is a disease process, a few of the disease-perturbed networks will modify the reflections of their cognate proteins. And every disease employs different combinations of disease-perturbed networks. So for each different disease, you get a unique fingerprint in the blood. Hence, you can use this to distinguish health from disease. And if you have a disease, you can distinguish which type of disease it actually is. And we've used these markers to do pre-symptomatic diagnosis, to stratify disease. That is, as you know, most diseases, such as breast cancer, aren't one disease. There are probably 5 or 6 or 7 diseases. And we've shown that we, for some of these diseases, can actually do the stratification from the blood. We can actually, in the case of prion disease, beautifully follow the progression of the disease, showing markers coming up from sequentially the 4 different networks. And then we can look at the response to therapy and recurrences. And in fact, as you heard, we started a year and a half ago a company called Integrated Diagnostics that's using exactly these strategies in connection with targeted mass spectrometry. And the results really have been absolutely spectacular to date. So, what about emerging technologies? How are they going to increase the patient data space dimensionality that we can begin to search? And let me talk about 4 big science-driven, technology-driven projects, and I'll talk a little bit about 3 of them, but not about the 4th. So one is the sequencing of families, which uniquely marries for the first time genetics and genomics and gives us enormous power in finding disease genes, and I'll explain that in just a moment. Number 2 is ISB, and Rudy Abersol, Rob Moritz have created something called targeted proteomics that for the first time give us real hope that we can push a human proteome project that's absolutely parallel to the Human Genome Project. And in fact, in the middle of February, we had our first meeting trying to convince funding agencies that we should do this. And I think the meeting was intellectually successful. Whether there's any money to pay for such a project is a different topic. We've developed a series of clinical assays, and I'll show you a few of those, that I think are going to open up completely new possibilities for what you can do with patients. And the one project I won't talk about, I call the Second Genome Project, and that is we have access to the world's 3rd largest data center, and we're going to use that to capture all human genome, fully sequenced genomes as they become available, together with their attendant medical records and phenotypic records and molecular and cellular data. And we're going to mine those for the predictive medicine of the future. And obviously you have to use all the latest high-level computing technologies and the cloud to do all the calculations and storage and that kind of thing. So family genome sequencing, we're doing it with a company called Complete Genomics that for us has done absolutely a superb job. We're doing it for about $6,000 a genome. This next year, we will do 615 complete genomes from a whole series of different families. But let me talk about what we did in the first study, which was published in Science last year. We looked at a family of 4. The parents were normal. The kids each had 2 different genetic diseases. And what we didn't realize at the time was the enormous power using the principles of Mendelian genetics would have in transforming our analysis. And it did so in many ways. Number 1, we could use it to correct more than 70% of the DNA sequencing errors. Number 2, we could use the family structure to identify immediately all rare variants. If 2 or more members of the family had the rare variant, it was a rare variant and not a sequencing error. You can never make that statement if you sequence a single individual. Number 3, we had the ability to reduce enormously the data space, the data search space for disease genes. I'll say more about that in a moment. And number 4, and this was the thing science was far and away the most excited about, we could for the first time measure the intergenerational mutation rate in a human family, and we demonstrated it's about 30 mutations per child. And of course, the really interesting question is, are all families approximately that have that rate of mutation, or will there be wide variations? And we have a whole series of other families, and we'll soon be able to answer that question. We don't have the answer yet. What we were able to demonstrate is by reducing the disease gene search space, we were able to say unequivocally that these two genetic diseases could only be encoded by one of 4 different genes, and by various ways we could make the proper disease gene assignment in a very straightforward way. So our feeling is now that for any simple Mendelian trait, family genome sequencing will give you the answer straight away. What we're doing now is we've sequenced 65 patients from families that have Huntington's disease, and we're looking for a modifier gene, and the modifier we're looking for is in those families, some members get it very early in their life, others get it very late in their life. So we'd like to find the gene that gets it very late and be able to turn that on for the other family members that have it. So it isn't a cure, but you could really change the quality of the life for many different people. And the analysis is almost finished. We should have data on that very soon. We think also we'll be able to take complex genetic diseases like Alzheimer's, and if we can do the proper kind of stratification, we can convert, that is, if we could say there were 10 subtypes of Alzheimer's, then each subtype would become equivalent to looking for modifier genes. And I'll show you how we're going to do the stratification after I tell you about some clinical assays a little bit later on. Now, the really key and interesting point, of course, is the amount of cost in sequencing has come down exponentially. So in 2002, it was estimated that the human genome sequence that was done cost $300 million. Today, we can get one that's much better data. Our error rate now is less than 1 in a million error rate, so it's spectacular. And that's a 50,000-fold reduction in cost. So my prediction is in less than 3 years, we'll be well below 1,000, and I'd say in 5 to 8 years, we'll be in the hundreds of dollars to do a genome. And the implication of that is you're all going to have your genomes done in the context of your families as a part of your medical records, and we'll talk about what that means a little bit later on. So the Human Proteome Project is one that ISB has really made 4 major advances to that I think really make it possible. And this obviously is a collaboration with Agilent and AB SciEx in an absolutely fascinating company, Chinese-American company called Origin. And what are the 4 things that we've done? Number 1, we've created a really complicated integrated software package called the Transproteomic Pipeline that essentially takes mass spectrometry data and evaluates its quality. Initially, when we created this, we threw away 95% of the data that we got from the literature, and it's improved somewhat, but I'll tell you, the quality is very country and lab dependent, so you almost know what the answers are going to be. So we put together, goodness, this little guy keeps popping up. We put together that transproteomic pipeline with the creation of a peptide protein database, the Atlas, that has about 4 million mass spectrometry spectra in it, and it's used by everybody in the proteomics world. And the technology advances are 2. 1, the creation of targeted proteomics using triple quadrupoles and MRM or SRM analyses. And the basic idea is that you can easily analyze between 100 and 200 proteins quantitatively in an hour if you put in radio-labeled isotopes as standards for the peptides that you analyze for each of those 1 to 200 proteins. So it means we can look at biological networks in a cohesive, integrated way within the limits of detection for the mass spectrometer. And to show you how powerful this is, Rudy Abersol and his colleagues have done MRM assays for all 6,000 some proteins in the yeast, and they can detect them from the lowest copied protein up to the highest copied protein absolutely beautifully. And that's exactly what we're going to be able to do for humans, because recently Rob Moritz has created MRM assays, SRM assays for all 20,000 human proteins. Not all have been validated, but they're there to be tested for the future, and that's going to be a key part of getting the Human Proteome Project started. Now, in the future, what we'd like to be able to do is identify 50 organ-specific proteins from each of your 50 major organs in the blood, and we'd like to be able to measure those 2,500 proteins so we could assess health as opposed to disease in a longitudinal fashion for each patient. So what we've done together with Caltech is really started working on a highly parallel platform, namely microfluidics protein chip assays. And right now, Jim has a chip that, one, can measure 50 proteins using ELISA assays from a fraction of a droplet of blood, 300 nanoliters, in a 5-minute assay with a dynamic range of close to 10 to the 6th. And we have these devices in the clinic at UCLA testing individual patients' response with melanoma and glioblastoma to drugs and antibody therapies by mere fingerprint pricks. And what's cool about that is you don't even have to go through IRBs to get fingerprint pricks. So the question is, how do we scale this from 50 proteins, and those are proteins whose ELISA assays have been created by industry spending gazillions of dollars to get them. And my argument is antibodies will never work for 2,500 protein measurements. So what we're doing together with Jim is using peptide protein capture agents, trying to develop these as protein capture agents. And the basic idea is the following. Jim has created a library of 10 to the 15th D-amino acid 6-mers. Some of them are branched. And what you can do is take the protein you'd like to create the capture agent against, and you can use it as a screen to get low-binding monomers. Then what you can do is take pairs of the monomers that are in their right three-dimensional orientation, and you can join them together to make a bivalent reagent with a chemistry called click chemistry that's been perfected by Barry Sharpless at Scripps. And you can actually even join at another level to get a trimer. And most recently, we've gotten tetramers. And with each mer you add on, you get 1 to 2 logs increase in sensitivity and specificity. And what is great about these reagents is they're rock stable, resistant to proteases. So Jim, with the first reagent he made, put it in the trunk of his car through the hot Pasadena summer, took it out in September and measured its affinity, and it was unchanged. So what's great about these reagents is they're things that we could send to the developing world, quite obviously. So we're very high on these reagents, but there are other things. Aptamers are making a comeback after a long drought, and so there are other possibilities as well. Let me just say that we've developed 16 different assays using various combinations of genomics and proteomics, and I'll talk just about one assay that I'm really excited about. Origene has expressed 12,000 human proteins in human embryonic kidney cells. And Origene has demonstrated that it can take 20 to 30 of these cells and place them on a glass slide and lyse them, and you essentially have a protein reagent there. And they've actually created chips that have all 12,000 of these proteins. And what they've demonstrated is if you look in autoimmune disease, you can pick up the autoantibodies beautifully. And my prediction is this is going to let us stratify autoimmune diseases in a way we could never think about doing before. Because if you take something like type 1 diabetes, I mean, with all the studies that have been done, there are 5 or 6 antigens that have been discovered, and I'll guarantee you the first time we look at patients with type 1 diabetes, we can double or triple that number just because you're doing the studies in a global rather than a one-at-a-time kind of way. And we could talk about any of those other assays. We're doing single-cell analysis. I'm going to tell you why that's going to be important in just a second. And we're going to be using induced pluripotential stem cells from individual patients. And that's really going to be important for stratification of disease. So just to give you an idea of how single-cell analysis is really important, we actually looked at 32 cells recently from human glioblastoma cell line. We looked at 24 different transcripts in each of those cells, and then we did a multidimensional analysis of those transcriptomes, and were able to demonstrate beautifully that those 32 cells fell into 3 quantized groups with a couple of outliers that were discretely focused and quite separate one from another. Now, we have no idea what those discrete groups do, but that they are different, distinct populations, there's no doubt about it. And with the Human Genome Atlas project, where you sequence whole tumors, in this case what you're doing is you're destroying the signal and you're increasing the noise. And I would argue these quantized cell populations really may be the key to successful therapy in the future. But anyway, we can use microfluidic platforms now to look at hundreds of transcripts from an individual cell. So what we're planning to do in the future is collaborate with a company called Cellular Dynamics, which has 2 unique capacities. One, we can send them 5 mils of blood, and from the white nucleated cells, they can every time get iPS cells. And iPS cells are really important because you can replicate them infinitely and make very large populations, and that's critical for a lot of things we'll want to do in the future. The second thing that they've learned to do is differentiate it to at least 4 major cell types. So they can differentiate them to myocardiocytes, 99% pure. All of these are 99% pure. Endothelial cells, hepatocytes, and most interesting, they can differentiate them to neurons. So what we plan to do in families that have Alzheimer's disease is to create iPS cells from all the members of the family, to differentiate those iPS cells so we'll have each patient's neurons in a test tube, and then we'll perturb those neurons with appropriate environmental signals, because each different type of Alzheimer's disease will have a different combination of disease-perturbed networks, and hence the environmental perturbations will be uniquely defining for each of the different types. This is all hypothesis. We haven't done it yet, but I'll guarantee it's going to turn out to be true, and we're moving along very quickly to do this kind of thing. So, the final thing is the analytic tools are obviously really going to be key, and it's late, so I'm not going to get into a lot of that. Let me just say that 1/3 of ISB is software engineers, computer scientists, theoretical physicists doing modeling, engineers doing computer science in one form. So we've created data pipelines that capture and validate and store and mine and analyze and integrate and model global data sets. And the most important thing I can say to young students is all software should be driven by the appropriate domain expertise problems. Don't invent software in isolation from the real problems. And here are a whole series of things we've done in the last couple of years that are absolutely related to specific kinds of problems, and I'm not going to get in that, but rather I'll leave you with a couple of interesting thoughts about computational challenges in biology. You have to realize data space for organisms is infinite, so you have to figure out how to formulate the perturbations that give you relevant insights into data space that can be informative. Number 2, high dimensionality of data, enormous signal-to-noise problems, and we've got to figure out how to deal with it. How do you separate all the biologies? How do you separate the technical kinds of things? Number 3, we need to have global measurements of all the different data types, and how do we deal with quality assessment and their challenges? And how do we integrate these multi-scale types of data to create predictive models and so forth? And one thing you should really understand in biology is how much information you need should be aimed at the nature of the problem that you want to solve. I would argue diagnostics in medicine needs infinitely less information than understanding biological mechanisms. So you want to design, these are really expensive studies, so you want to optimize your studies to get what information you need, but not to be excessive about it. And finally, one of my favorite points is there's no such thing as a pathway. There are only networks. And I'll tell you, I've seen time and time again people make major mistakes in talking about pathways because it focuses their thinking in linear dimensions rather than in multi-dimensions. So as a student, that's really a key point. All of these things bring us to this thing called P4 medicine. And let me just give you my 10-year view of where we'll be with some of the dimensions of the 4 Ps. So predictive. I predict that in 10 or so years we'll all have our genomes done, together with some environmental information that will give us enormous insights into the future trajectory of our health. And we can advise patients in ways we've never thought about advising them before with regard to wellness. And wellness is going to be a key theme in P4 medicine, as you'll see in a moment. The other prediction that I would make is, I think easily in 10 years, we'll have a little handheld device that can prick your thumb and take that droplet of blood, make 2,500 measurements, send it via wireless to a server and analyze the information and come back to you and your physician with, you're fine, do it again in 6 months or whatever. And I think this is going to be enormously important, not only for detecting the onset of disease, but for reinforcing the things that optimize wellness as well. What about personalized? Well, we differ by 6 million nucleotides on average, one from another. When I was in medical school, I could never understand why physicians would say, let's take and average these patient populations and we'll say everybody that falls in the extremes is sick. And I could never understand it because of genetic variation. And the fact is, every patient is going to be their own control for health and disease. It has to be, that's the essence of what personalized medicine is. But, just think about it. You'll all have billions of data points. We'll be able to sculpt with exquisite sensitivity the dimensions of health and wellness and predictions about the future for each of us. And even more, we'll have, in America, 360 million people with billions of data points, and that will give you the resources, again, to mine for the predictive medicine of the future if, and this is really a big if we can get around all of the societal constraints and IRBs and all those things. I think it's about 50-50 that all of this mining for the P4 medicine of the future will be done in China. Because Europe and the US have exactly the same impossible constraints at this point in time. Unless it gets fixed, we're going to lose out in a big way, like we've lost out on some other kinds of things. The preventive side of things is, on the one hand, this new approach to identifying drug targets, using drugs to perturb networks to make them behave more normally. On the other hand, for the first time, we're going to really understand how to effectively turn on innate and adaptive immunity and create vaccines that are really going to be effective for malaria and AIDS and all the things that I remember having an enormous argument with Tony Fauci 10 years ago, and my argument was, take the billions of dollars you're going to spend on AIDS vaccines and put a small fraction of it into systems approaches to understanding the immune system. They did that this year. So they're only 10 years off, but at least they're getting started. They have wasted billions of dollars giving it to companies that essentially did the vaccines just exactly like Jenner did in 1796, quite frankly. I mean, there have been a couple of exceptions, but the exceptions didn't work either. So participatory deals with social aspects. So how do you educate patients about the potential of this? How do you convince a skeptical physician? How do you how do you inform the medical network that's going to have to carry out this? What you have to realize is that P4 medicine is a real revolution, and it's going to require changing the structure of our healthcare delivery system in some really major ways that people are going to feel uncomfortable with. And I'll tell you later how we plan to approach it, in one way anyway. So let me give you 4 societal implications. Number 1 is this is going to force, in the next 10 years, it'll force every sector of the healthcare industry to totally rewrite their business plans. And the really interesting question is, can old companies learn to do new things? And I think in most cases, they're going to really have trouble. And this is going to be true of a lot of drug companies. There'll be enormous opportunity for people that are at the leading edge of this to create new companies like Integrated Diagnostics. Number 2, this is going to lead to an incredible digitalization of medicine. And it's a digitalization in 3 different dimensions. So number 1, how are we going to deal with these billions of data points? Number 2, we're actually going to be able to get actionable information from one molecule, one cell, one genome, any unit of information in biology. That's what this quantized digital kind of meaning. And number 3, very soon we're going to have iPhones for health that will be recording hundreds, if not thousands, of our own data points and feeding back continuous information on how our multi-systems are responding in various ways. This is already starting to happen with Eric Topol at Scripps in really interesting kinds of ways. So I would argue this digitalization is going to have an impact far greater even than the digitalization of information technologies and communication. Number 3, I argue that P4 medicine is actually going to turn around the ever-accelerating costs of healthcare, and they'll come down to the point that I think we'll be able to export it to the developing world. That is, P4 medicine will be the framework for global medicine. And I think this for 5 reasons. 1, diagnostics will let us stratify disease and make ideal impedance matches against the appropriate drugs. Number 2, we're going to have a completely new way to identify drug targets, and hence a much more efficient way of creating drugs. They're not going to cost $1 billion anymore. And we can test them real time along the way in abort studies at an early, because more and more we're going to be able to do studies with very small groups of patients. And the reason we can do that is if you take 40 patients and you have a 98% success rate, you've got all the power you need to convince the FDA to make that a drug. And Genentech did that for the drug called Herceptin for breast cancer. Number 3, the benefits of wellness, I think, are going to be absolutely staggering. Number 4, these technologies that are exponentially increasing in measurement potential and decreasing in cost are really, really going to let us sculpt the dimensionality of individuals in really interesting points. And it's going to be done inexpensively. And then number 5, I think there are going to be medical advances in stem cells, neurodegeneration, aging, vaccines, and the like that really are going to be transformational over the next 10 years. And finally, I'd make an even more positive argument. The nations that really adopt this, it's going to be a source of enormous wealth. 1, it's going to reduce the cost of health care significantly, but on the positive side, in an information-based economy where people can stay well, you increase enormously the productivity. And economists can do calculations, and the calculations easily are in the hundreds of billions of dollars for that kind of thing. And then finally, it's going to spawn 2 categories of new industries, a wellness industry on the one hand, and the health industry on the other hand that'll be totally different from the health industry we know here. So all of those are enormous opportunities for creating wealth. So the challenge of P4 medicine is twofold, technical and societal, and the societal is going to be by far the bigger challenge. So how do we go about really making this happen? Something we've really thought a lot about at ISB. We're setting up key strategic partnerships to bring P4 medicine to patients. And let me tell you just about 2 of those. One is an agreement we made about 2.5 years ago with the state of Luxembourg that gives us $20 million a year for a 5-year period to attack several of the most fundamental problems in P4 medicine. In return, we do a lot of things for them, and I'd be glad to talk about those, but I'm not going to talk about it here. Number 2, we've created a nonprofit institution called the P4 Medicine Institute in partnership with Ohio State. And the idea here is that that institute is now creating a network of healthcare centers that will use these assays we've developed at ISB and use them in the context of pilot projects. Because I'm really convinced, to a skeptical world, the way you convince them is you show them that you can do things that never could be done before. So our first 2 projects are a wellness project. So we're measuring for the first time, we're developing for the first time metrics for measuring wellness. Be glad to talk about those. And number 2, we're going to look at heart failure, which is a major problem that really has been intractable for a long time. So the essence of P4 medicine then are 1, quantifying wellness and demystifying disease. And of course, it's these major societal opportunities that we talked about, the economic opportunities, the digitalization of medicine, the turning around the costs of medicine, and finally, the creation of wealth and so forth. So let me end up with 4 final comments. So the first is about students and how we are improperly training them. This is one of my favorite New Yorker cartoons, and it says, "Never ever think outside the box." And the real question is, how can we change our educational process so our students will really think outside the box automatically and never, as most of them do? And of course, a part of this, I would argue, is key cross-disciplinary training. I think this is one of the big revolutions coming in modern biology, and it seems to me, from what I've heard, some of you here at Iowa State are in good shape in that regard. Point number 2 is big and small science, and by big science I mean integrated systems approaches to big problems. As you know, at NIH there is enormous pressure to do away completely with big science. Anytime budgets get tight, all of the small scientists and the bulk of people at NIH are small science-oriented people. In fact, I was just on a committee set up by General Medical Sciences to review the 4 major GLUE grants, big science grants, and it was really interesting to see the composition of the committee and how they ended up arguing in various ways. But I hope it's going to come out to a good end. We'll see. But the really important point is that at NIH, I would guess 5 or 6% of the dollars go to big science, 70 or 80% of the dollars go to small science. So keep that in mind as we're making these arguments. And I would argue there are many problems that cannot be approached by small science at all. They're just too big. They need integrated, cohesive, well-led efforts to attack them. And the other point I'd make is that there is enormous synergy between small and big science, because big science creates the fuel for enormous opportunities for small science. We've had terrific interactions with a lot of people in small science. A final point, a third point, is the idea that all of these paradigm changes that we saw required new organizational structures. And that's certainly going to be true of P4 medicine, and I wonder what other ones are going to be required. And I will tell you what my plan is. So I was at the 150th anniversary of MIT about 2 weeks ago, and Eric Lander and I gave our talks, and then we're on a panel, and someone asked me the question, well, how are you actually going to really bring this P4 medicine to the world? And I said, well, we're going to do pilot projects in this network of institutions that we're setting up in the US, and then what we're going to do is we're going to go to Luxembourg, because I know the three key ministers, and they've all agreed that if our pilot projects are successful, we will bring P4 medicine to Luxembourg. So it's 500,000 people. It's the highest per capita income in the world, and it's a single-payer system. So it's an absolutely ideal test model. If it really works there, then maybe this heterogeneous mess of a system we have in the United States. So Eric Lander got up immediately, said, you know, this is treason. Why are you sending it away? Why don't you do it here? So I said, Eric, I wrote a paper for the Obama transition team that discussed P4 medicine and healthcare. And I said, I never even got an email back of thanks. So if you have real connections and can get something going, let me know. But so anyway, but that's what we plan to do. And the final point I would make that I've really thought a lot about, I don't know how many of you read Tom Friedman's book on The World is Flat, but his point was essentially that there is a globalization of education and economies, and I would argue there's a fascinating globalization of science that's occurring too. And what this allows us to do for the first time is to ask the question, who are the best scientists in the world to attack really hard problems and bring intense focus on solving them? Because getting the best really makes a difference rather than maybe just somebody who's next door to you. So that's really what we've tried to do in the strategic partnerships I talked about and the ones that will be in the process of setting up in the future. But I think it's really also interesting from the point of view of the U.S. government isn't going to fund big science except at a very, very low level. And I'd say $3 to $5 million a year is a low level, particularly if you have to have 10 people involved in the grant, and by the time you divide it, it's a glorified R01. But if you set up these strategic partnerships, if you can get $20 million a year for 5 years, that lets you do things that are really nonlinear with regard to future possibilities. So lots of people have been involved. I'm not going to go through and name beyond the ones that I've already named, but it's been a great partnership with many different people on these things. So thank you very much. So I'd be glad to answer questions.
I don't know how to make that microphone work, but I have a question really directed towards prenatal applications of these technologies. And that is not something you've touched on, but all the medical implications are for the postnatal individual. And this raises a number of enormous concerns. I work on rare fatal neuropathic disorders in children, and I'm going to be out of a job in 10 years apparently because we'll be able to screen for those diseases in ways we haven't been able to. And that may be a benefit, but I can see application of this technology that is enormously troubling in that sphere. Could you comment on that?
Yeah, I could comment on that. I mean, that's curious. We have just begun to talk with Children's Hospital in Seattle about a strategic partnership that will be looking at this whole problem of maternal and fetal infant health and so forth. And so what we're setting up to do actually is to look at several major fetus-threatening conditions and to, from those placentas, get the placenta-specific proteins that are placed in the blood, so that we will be able to follow those kind of conditions from conception all the way through to birth delivery. So I think this is an area that almost nothing has been done, and it's an incredible area of opportunity, and I think many of the technologies that we have here would fit into that beautifully, but especially these blood protein assays that I think could let you assess directly the placenta and indirectly the mother and the fetus. And in fact, we may be able to look at maternal and fetal blood proteins that would be interesting in this regard too. So I think there are a lot of things that could be done. I think that is really an important area. It's actually an area that the Gates Foundation is really getting interested in, so we're hoping to get major funding to do this kind of thing from them. Other questions? Yes?
Would you agree that what's missing from the advocacy of this technology is patient testimonials? Are there patient testimonials that we could hear or read about today? If not, how far in the future are they?
Yeah. So the question is, are there patient testimonials that are missing from this? And again, understand that we're just getting started in these things, so there haven't been any chance for patient testimonials. But I'll tell you where there are patient testimonials that are really interesting is in doing genome sequence analysis. So companies like Navigenics and 23andMe now have quite avid followers that are really excited about the potential of using genomics to be able to assess disease possibilities. If you're normal, you may be disappointed. There may not be much you can see. But, for example, 23andMe now has identified more than 6,000 family members from families that have Parkinson's disease. Now, the CEO of 23andMe has a husband who is susceptible to Parkinsonism, so it's a particular interest. But these families are enormously enthusiastic about doing whatever it takes to learn more about it. I mean, I think, the question of whether data and making your data are available, I mean, as I look at what's being done in social networks, I really come to the conclusion that if we wait for a little while, that isn't going to be a problem. Because what they put out on FacePage, my goodness, is far more than your genome sequence and things like that. But seriously, what we do have to do with these making patient records more accessible so we can do the mining is make sure that there are laws that protect you against insurance companies, against employers, and actually, frankly, against your family to a certain extent too. And those are slowly being put in place, not very effectively, but slowly. Yes?
Yeah, I just have a comment about your radio analogy. I mean, in the case of a radio, you know that the nuclear excitation levels of the copper nucleus of the wire are irrelevant to the working of the radio. So you can focus on, what is really essential. Okay, now in the case of biology, I guess we're just not that well developed. And if you have this tidal wave of information, which is getting cheaper and cheaper, is there the danger that you get sidetracked looking at, completely irrelevant dynamics for the problem you're interested in?
Well, I think anytime you're dealing with large data sets, the real problem is signal to noise. We still don't have very good ways for dealing effectively with that. And I think that's the big one that we have to get over. But sure, there is always, in any complex process, a biologist can get lost in the trivial details. There's absolutely no question about it. So what's really key is to think about the deep principles and try and keep yourself on the right track of where you want to go and what you'd like to solve. But yeah, in biology, as that Rube Goldsberg cartoon showed, they're infinite detail. You can get fascinated by all sorts of little things. And frankly, this is what happens to a lot of people who do small science. They do things that aren't really very important because they have no broader context from which they can make judgments about how important that is. One of the questions I asked on this committee was, look, we're having a review of big science now to see how well it did in these things. Has anyone ever reviewed small science? For example, has anyone ever reviewed the fact there must be 600 labs that are studying P53 and they're all doing almost exactly the same things, some better, but is that the best way to do things? So, it's a very complicated, multidimensional equation. So, yes?
So I'm just curious, it's my understanding that this integrated approach to complexity and this whole systems biology approach is going to give us a much more holistic, wider picture of networks and components of the biological system that we have over time by studying individual components. But do you think if this trend increases in science, if this approach, this cross-sectionary approach increases?
I'm sorry, you'll have to speak up a little bit. Couldn't hear the last part.
Do you think this cross-disciplinary, multi-pronged approach to science and biology in general is going to take us back a few findings too? Because, as you said, small science, small discoveries might have been just noise. One gene expressing at a higher level in the system by treating it with some stress would just have been noise that was reported in science. So do you think this multi-pronged systems biology approach is going to refute that finding someday?
He's wondering if some of the existing scientific literature would be negated.
Well, I think it's less negating than explaining the results that they find. And the important question, I think, isn't saying you're wrong, because I suspect good people aren't going to be wrong, but is it important? That's the really important question. Should we be studying this kind of thing or not? So I don't think they'll negate anything. I think they'll just put it in context. Small science. Yes?
Would you comment about the potential barriers that gene and protein patents may pose for P4 medicine in terms of developing and implementing new therapies?
So, will patents pose a barrier for P4 medicine? The interesting point is, at this point in time, I don't think so, because a lot of the things that we're thinking about are so new and so different. And let me give you an example. When we first put a patent in on organ-specific proteins, probably 8 or 10 years ago, there was nothing in the patent literature that was even remotely close to that. So what's been very attractive is these really new ideas have a lot of space for patents to be expanded around. Now, where things can get complicated, obviously, is with genomic information, where there's an enormous amount of data out there. And how exactly all of that is going to tie into being able to do whole genome sequences. What I think is going to happen in the end is companies that own individual genome sequences are not going to be able to sue 340 million people because they had their sequence of the BRCA1 or 2 gene done. So I think just by sheer force of the magnitude of what's going to happen, those kind of, that won't be a problem. I think what we are going to see more and more are what I call systems integrative patents, where you put together collections of information that are uniquely defining for disease state. And there's not anything like that out there, except maybe a few multidimensional transcript studies, genomic health, and things like that. But there are much more sophisticated ways you can do a lot of those things. And I think even integrating different kinds of information together is really going to be informative in interesting ways, and we're just figuring that out right now. Other questions? Yeah.
So you mentioned that there are 4 different paradigm shifts that you noticed where new institutes or companies or these organizations had to be created, and you were involved in a lot of them. So what were the giveaways for you? What made you decide that the whole system isn't working and we had to get a new one for that?
Well, I think the giveaway in a paradigm change for a person who's pushing the paradigm change is utter conviction that this is the way it's going to be and this is the right way to do it. And fortunately, I've always had that conviction. Now, I'll tell you, just to give you an example, when I went to Caltech in 1970, I told my chair I was going to spend 50% of my time doing engineering, developing technologies. And he came in after 3 years and he said, I'm urging you in the strongest possible terms to give up all of this engineering. And I said, no, I wasn't going to do it. And I got tenure 2 months later, so it didn't have anything to do with tenure. And 20 years later, I said, why did you say that? Do you remember that? And he did. And he said it because all of the senior professors in biology at Caltech felt it was unseemly to have engineering and biology And he said, what they suggested I do is move you to engineering. And he said, at least I didn't make that suggestion. I said, yeah. So, I think you just have to be really convinced that what you're doing is right, and you can be convinced, and sometimes you're wrong. But on the big ones, it's better to be right. There's no question about that. Yeah. Over there. Well, my conviction, so the question is, can we use systems engineering to approach really difficult problems like cancer, where you go through multiple events and so forth? My feeling is the key to that is single-cell analysis. I'll guarantee you that's going to give us enormous insights into the populations and the transitions and the quantized groups that actually carry out some. So I think what we have to be able to do, what we'd love to be able to do in the next couple of years, is to set up a microfluidic device that can do the entire transcriptome for 1,000 cells in one hour run. Then you could really begin to take on problems like that. But I think it's again getting down to the modules of information, and one of the key modules is the cell. And when you average the populations of cells, you lose the signal. I mean, that's just clear again and again and again. So that's the kind of way we're thinking about it. Was there a question? Yes?
I'm really impressed with the idea of genome sequencing of family. We're biological and adoptive parents. Our son is adopted, and he would like to find out who his biological parents are. The states will not let that information be released at all. He does not know who his parents are. So is that one of the social problems that you're talking about that's going to be resolved to do?
Well, there are a whole bunch of social problems. Now, that would be something that could be solved by going around state bureaucracies. But I'll tell you, so we're thinking of setting up a strategic partnership with Saudi Arabia to look at the royal family, and they have simple genetic diseases and complex genetic diseases. One of the things that they're terrified about is sexual incest. And I mean, these analyses will say, gee, you're really the daughter of this guy over here and not the daughter of this guy over here. And so we haven't quite figured out how to get around that one. But there are problems like that. And in fact, there was a recent paper in Lancet that showed large-scale GWAS studies that absolutely identified several cases of incest that were utterly clear. So, those are the kind of things you have to be sensitive about. Yeah.